In this video, we'll take a look at glomerular filtration rate and its homeostatic regulation. Like all homeostatic regulated variables, it'll be regulated by intrinsic and extrinsic mechanisms. As a quick review, intrinsic mechanisms are also known as autoregulation, as they come from within the organ or system itself. Extrinsic mechanisms are ones that come from outside the organ or system, invariably include hormones, the autonomic nervous system and its role, and potentially other things that may affect, in this case, glomerular filtration rate, or what we call GFR. So we'll go through each one and take it like we did in the past, and we'll kind of look at the different factors that affect glomerular filtration rate. So let's start with the intrinsic regulation of GFR. That is also known as autoregulation. And there are two intrinsic regulatory mechanisms, and they are somewhat similar to the mechanisms that regulated TPR that we talked about in the cardiovascular section. One of them is actually exactly the same, and that's the myogenic mechanism. So remember, myogenic, myo was muscle, genic means it comes from. So the myogenic mechanism comes from muscles. The arterioles can alter their own diameter to help maintain flow. So that's the myogenic mechanism. And then the tubular glomerular feedback mechanism we already talked about with the macula densa and things like that. So let's look at a little more detail about each one. The myogenic mechanism is an autoregulatory mechanism that helps to maintain glomerular hydrostatic pressure. So this is, again, the arterioles can change their own diameter in response to pressure or stretch within the arteriole. So if we increase the blood pressure, that causes an increase in stretch. That means we get more constriction right here, and that increased resistance will decrease flow. So the flow is maintained to the original number, and that maintenance of flow then should keep GFR the same because the glomerular hydrostatic pressure is maintained. So arterioles have the ability to do that, and it's not any different than what we saw in a regular arteriole, the myogenic mechanism, for the general circulation as well. The second mechanism we've mentioned before in a previous video, and that's the tubulo-glomerular feedback mechanism. And that involved the macula densa and JG cells and the whole juxtaglomerular apparatus where the distal convoluted tubule crosses the afferent and efferent arterioles. And remember that the macula densa contains a number of different types of receptors. So it's this purple area here, although it meanders kind of all over the place from what I understand. The types of receptors that we find here are osmoreceptors, so they measure the osmolarity of blood, chemoreceptors that look at specifically the chemical composition of blood, and mechanical receptors for stretch pressure in the kidney, perhaps even flow. And so what they do is they respond to changes in the tubules, and they actually also monitor the arterioles. And based on changes in the tubule solute or flow or pressure or stretch or chemical or osmolarity, whatever it is, they can signal the arterial smooth muscle, and that can change diameter to change things back. So as an example, if the osmolarity in the distal convoluted tubule was high, right? So meaning that it had a lot of dissolved particles. So if it's got a lot of dissolved particles, it could signal that there's not enough water there. And by stretching the afferent arteriole, that would increase the flow of fluid into the glomerulus, which would then give us more filtration pressure, which would give us a higher GFR, which would maintain a normal flow coming out. So the idea is that we can maintain this normal flow based on that. So the fluid flow through the tubules influence the GFR. If we decrease flow, then we get an increase in osmolarity because we'll have more dissolved particles. And the macula densa, right, will cause afferent arteriolar dilation, which then increases GFR. Remember, if the afferent dilates, if we decrease resistance of the afferent, we get more blood into the glomerulus, which means we get a higher filtration pressure and a bigger filtration. And the opposite is true if we go the other way. So the tubular glomerular feedback mechanism is sort of like the idea with the TPR, but a little more complicated with the chemical hypothesis. So this is like the chemical hypothesis on steroids, right? And it doesn't involve just chemicals, although there are certainly chemoreceptors there. But there's also osmo and mechanoreceptors that also help play a role to keep GFR constant. So those are the intrinsic mechanisms. The extrinsic mechanisms, we'll do the easy one first. And the easy one is the ANS, the autonomic nervous system. And the reason why the ANS is easy is a couple. So one, and again, this is just our convention, notice we have two red lines going to the efferent and afferent arterial, one to each. Now, if you remember from previous videos, and you may not, we've sort of kept this pattern. The red neurons represent the sympathetic nervous system. And so the kidney, like many structures, is only innervated by the sympathetic nervous system. So first of all, we don't have to worry about any parasympathetic stimulation changing what happens in the kidney directly because there are no parasympathetic fibers that go there. We do have sympathetic fibers that go to the kidney. You can see here in the baroreceptor reflex drawing. Okay? And the idea is, if you remember from the baroreceptor reflex, if we increase the sympathetic system, we increased blood volume. And so I actually had a question on that from a student about how that worked. But the way it basically works is the increased sympathetic system causes constriction of the afferent arterial. Okay? So we mentioned this earlier too. Even though both the efferent and afferent both have sympathetic fibers that go there, the stimulation affects the afferent more. The reason is the afferent has more what we call alpha-1 receptors. Remember from the ANS system, the alpha-1 receptors are the receptors that cause constriction in the arterioles. So if we didn't do that anatomically, remember it's the receptor that gives the effect, that was one of Cannon's postulates basically, then we would stimulate both the afferent and efferent, and they would both constrict, and the constriction of the afferent would decrease GFR, and the constriction of the efferent would increase GFR, and we'd really probably have no net change. So the way our body has dealt with this issue is we just have a lot higher innervation of receptors in the afferent arterial, which means the same stimulation that goes to the efferent goes to the afferent, but it has a greater effect because it has more receptors. So when we talk about the effect of sympathetic stimulation, we often just talk about the afferent because of its greater density of receptors. So it will have a greater amount of constriction than the efferent, and it's the one that's playing the major role in terms of regulating GFR sympathetically. So if we increase the sympathetic, then we get constriction of the arterial. We get less blood going through the capillary. That decreases glomerular hydrostatic pressure, and that decreases GFR. At the same time, the sympathetic system directly goes to the JG cells, remember the juxtaglomerular cells, and they release that renin directly into the bloodstream. And renin, we'll see in a little while, activates a hormone called angiotensin, eventually to angiotensin 2, but it's the first step in activating what they call proangiotensin to angiotensin 1. So that's the other effect the sympathetic stimulation has, and that hormone that we eventually activate will do similar things in the blood vessels as epinephrine. It would cause constriction. So that's the sympathetic stimulation and its direct effect in terms of decreasing GFR and increase in the release of renin. So by far the most complicated portion of this is the extrinsic regulation by hormones. And we can look at two different types of hormones, and it makes sense, again, if we think about it this way, because when we've talked about homeostatic regulation a number of times, we've mentioned that one of the key components of it is the ability to have antagonistic control. It means we should be able to go back to the set point. Remember, the set point for GFR in most people is about 125 mils per minute. So if we went too high, we'd be able to bring it back lower. If we went too low, we'd be able to bring it back higher. So that extrinsic control also happens in hormones in this case. We have water conservation hormones, hormones that ultimately help us to conserve and retain water. And we have a water excretion hormone that helps us get rid of water. Now, interestingly, from an environmental point of view, it's generally much more difficult to hold on to water and find it in the environment than it is to excrete it. So not surprisingly, from that point of view, we have 3 different known water conservation hormones and only 1 known water excretion hormone. So we'll look at the ones that help to conserve water first. So remember, the long-term regulator of blood pressure is blood volume. So these first 3 hormones that help to increase blood volume also help to increase blood pressure. ADH, as I said before, is one of our major water conservation hormones. So one of the main effects of ADH is to maintain blood volume because it enhances water reabsorption. ADH has a couple names. It's got a number of different things we call it. First of all, its name most often used is called antidiuretic hormone. That's where the ADH comes from. The reason why we call it antidiuretic hormone makes a lot of sense when you think about it. The diuretic portion means that people would pee more, basically. So antidiuretic would mean that people pee less. If we pee less, we retain water. So that's one of the jobs of antidiuretic hormone. So I call it ADH. The other name is called vasopressin. Vaso refers to vessel. Pressin is pressure. So it's a very potent vasoconstrictor. The vasopressin is sometimes just called VP, or some people even call it AVP for arginine vasopressin. Historically, vasopressin was discovered as a neurotransmitter, while antidiuretic hormone is a hormone. Actually, technically, it's a neurohormone. We'll see in a second. So those are the 2 names. And basically, if you can remember the actual 2 names, then you can remember its 2 major functions. So for these hormone discussions, we're going to look at a couple things. So first of all, we're going to look at the source. So where does ADH come from? Where do we get antidiuretic hormone from? It's a little complicated. It's actually produced in the hypothalamus. We won't worry about what part of the hypothalamus, but it's produced in the hypothalamus in the cell body where the rough ER is, right? And then it's transported through fast axonal transport. We talked about that earlier in the semester, to the synaptic end bulb where it's released from the posterior pituitary. And since it's made from nervous tissue, it's technically a neurohormone. It's not really any different than a neurotransmitter, except it's released into the bloodstream directly and not into a synapse. Its release is controlled by a couple different things, and one of them is the osmolarity of blood. Now, if you remember from anatomy, the hypothalamus is an area in the brain that is not protected by the blood-brain barrier. So the hypothalamus actually sees the blood the way it should be in terms of not being protected by that blood-brain barrier. So the hypothalamus is sort of the main portion of the brain that controls a number of these processes like temperature and osmolarity and a number of other things because of the lack of the blood-brain barrier. So it measures the osmolarity. If you remember back again earlier semester, we talked about the normal osmolarity of human fluid is about 280 milliosmoles. So if your osmolarity goes up, it means your blood is becoming more and more concentrated. And so the idea would be we would increase more antidiuretic hormone, and that would help us to maintain more blood volume, and that would help dilute the osmolarity of blood back down to normal. The same is true if it went the other way. If osmolarity decreased, it means that we have a lot more water in our blood, and so we would decrease the ADH, which would reduce the blood volume and help concentrate our milliosmolarity back up to what it was for normal. So I mentioned the basic actions. The first one, again, is antidiuresis, right? So it prevents you getting rid of water, so it helps to stop pee, so it increases blood volume. That's its major mechanism of action, is to retain water. When we retain water, we also increase blood volume, and then we also increase mean arterial pressure, so we help to increase blood pressure. Its minor, as I mentioned before, its other name, vasopressin, is vasoconstriction, and that increases TPR, and that also helps to increase mean arterial pressure. So the two targets, one, probably should switch these, is the distal nephron, and really the distal convoluted tubule. And when we talked about the functional anatomy, we talked about principal cells being located there. There's a few in the collecting duct, that's what that CD is. But most of the principal cells are in the distal convoluted tubule. And what happens is there's aquaporins in the cells within the tubules. Aquaporins, one of the better named things in physiology, because aqua means water, porin is whole. And so it increases water reabsorption. Probably the easiest way to think about it is over here in the yellow, that's our ultrafiltrate, right? And if we don't take things out of here, it's going to go out as urine. Here's our blood vessel over here on the right side. And then here is the interstitial fluid. So what happens is ADH actually opens up, and you can just think of it as a channel that goes from one side of the membrane to the other. And water would go through this and go all the way to the other side and get reabsorbed in the body. When there's no ADH, this gets blocked basically, and the water stays in the ultrafiltrate and then eventually goes out as fluid. So ADH opens these aquaporins and helps the reabsorption of water. The minor mechanism of action, the vasoconstriction, that's just due to this one, so we'll kind of crisscross those arrows, to the vascular smooth muscle and vasoconstriction. So it's the smooth muscle in the arterioles especially that constrict and cause vasoconstriction to increase TPR and eventually increase mean arterial pressure. So those are the targets that we see there as well. Okay, so the first one is known as ADH or. So there are a few other things that we can mention about antidiuretic hormone ADH. It is increased by pain, stress, and trauma. Also morphine, nicotine, and other drugs will also increase the secretion of antidiuretic hormone. Interestingly, it's decreased by alcohol consumption. When I was in graduate school, I had a professor that said, drink a 6-pack, pee a 7-pack. And the idea is the alcohol has a direct effect on the hypothalamus, which then decreases the release of antidiuretic hormone. You can have increased or decreased release of antidiuretic hormone for a variety of reasons. But one of the things I just want to mention is the hyposecretion. And so let's go on to the next water conservation hormone, and that's called aldosterone. A couple things about aldosterone that you should probably kind of key into. Okay, so let's look at the next water conservation hormone, and that would be aldosterone. Aldosterone gives you a clue a little bit by its name, not always, but in this case, often, and this is one of those cases, when a chemical in the body ends in ONE, it is typically some sort of steroid hormone. Yeah, there's other, GEN, like estrogen, can do that as well. But when you see the ONE, you can say, okay, it's probably a steroid hormone. Aldosterone, because it's a steroid hormone, and you can see that here in the middle of this cell, this blue cell, affects the rate of what's called transcription and translation. And basically, it's how do we read DNA with RNA, and then how do we take the RNA and produce a protein from that. And because that takes a while to do those things, physiologically, at least in terms of time, that relatively speaking, steroid hormones tend to be significantly slower acting than the other types of hormones, the non-steroid hormones, the protein hormones, basically. So aldosterone is made in the adrenal cortex. So if you look here, you see the adrenal cortex. The adrenal gland has two basic layers, an outer layer called a cortex and the inner layer called the medulla. You might remember from your anatomy course, you might have discussed the three layers of the adrenal cortex, the zona glomerulosa, fasciculata, and reticularis. The aldosterone is made in the outer one, in the glomerulosa. And its job, right, its basic action, is it retains sodium, which then allows us to retain water, which then allows us to retain blood volume, which then allows us to increase mean arterial pressure. So, like all of the water conservation hormones, it increases blood volume and blood pressure in some form. The targets are the ascending limb of the loop of Henle. So if you remember when we talked about the functional anatomy of the nephron, the ascending limb of the loop of Henle is permeable to solutes, but impermeable to water. And what we have in that area, in this case, and one type of cell we'd be interested in, is one that has sodium reabsorbers. And basically the sodium reabsorbers are, their job are proteins that reach into the tubular lumen and pull sodium out. So instead of peeing out sodium, right, we pull sodium back into the body. And because we pull sodium back into the body, then water eventually follows, and we help to retain water, which then increases blood volume and increases blood pressure. But it takes a while to affect these sodium reabsorbers. We increase the number and or the activity of them, so we can actually make them work faster, and we can increase the actual physical number of them as well. Ultimately, it takes, in some cases, up to 45 minutes to have aldosterone released, and then before you have the full physiological effect. Because you do have to affect, like I said before, how we produce a protein in a cell. So those are the targets. What's kind of interesting is what increases the amount of aldosterone. And some of it makes sense, but in some cases, it'll take a little more explanation. So one of the things that increases aldosterone is a decrease in sodium. That makes total sense, because what it does is it helps us to increase sodium in our body. So if we had low sodium, it would make sense that we would increase, as you kind of see down here at the bottom, the increase of reabsorption of sodium. Also an increase in mean arterial pressure. So if your blood pressure, your BP goes down, right, it makes sense that one of the basic actions is to increase blood volume and blood pressure. It would make sense that low blood pressure would trigger this. So that makes sense. One of the things that doesn't initially make sense to most people is an increase in potassium. Why do we care about an increase in potassium? Well, In order for the body to maintain a charge balance, if we retain a sodium or get rid of a sodium, whatever it is, then we have to balance that charge. So theoretically, that sodium was being released by the body. Okay? It was in the ultrafiltrate and it was going to get peed out, basically. So if I retain that positive charge, I have to retain some either negative charge to balance that, or pee out some other positive charge to balance that. And that's called isoelectric equilibrium. So maintaining the same electrical charge in our body is an important consideration, often not talked about in any course or anything, but it is an important consideration. So basically, if we have a high potassium level, then what we're going to do is we're going to retain sodium and spit out potassium in its place. So if you see here, in the yellow on the left side of this figure, it says potassium secreted to balance sodium absorption. So that helps maintain the balance. We can also do that with, not just potassium, but any other ion. We can either retain a negative, an anion, along with the sodium, or we can get rid of a cation along with the sodium. So acidosis also does the same thing, which makes sense when you think about it. So acidosis means you have an excessive amount of H+. So if you sit down here in the bottom right, hydrogen ion. So if I want to get rid of hydrogen ion, then I can increase the aldosterone, right? And that would increase the sodium reabsorption. But at the same time, to balance that sodium reabsorption, we would get rid of the H+. And as we got rid of the H+, that would allow pH, which was, if it was acidotic, too low, to go back up. So aldosterone has some other mechanisms that don't make sense unless you think about them for more than a second or two, but ultimately don't seem too difficult. So that's aldosterone. The last hormone for the water conservation class is angiotensin II. Now, angiotensin II is by far the most potent vasoconstrictor we have in the human body. Meaning, if we have the same amount of epinephrine or adrenaline or antidiuretic hormone or, a number of other oxytocin and other vasoconstrictors, angiotensin, if we had them all in the same amount, would get much more vasoconstriction than any of the other ones. So the actual hormone that has by far the biggest physiological effect is angiotensin II. We'll kind of mention how that is. So what's the source? So notice we see multiple sources of these. We'll kind of look at it this way. So the first one word you see is liver. So if we come over here in the figure, we'll see liver. And the liver is constantly producing this precursor of angiotensin II that is sometimes called angiotensinogen. So if it's got the word ogen at the end, it means it's an inactive form. Or sometimes we put the word pro in front of it, like proangiotensin. It just kind of depends on who you're talking to. Either one is appropriate, although some people prefer using this one, angiotensinogen, more than they do proangiotensin. But they basically mean the same thing, an inactive form of this hormone. Okay, so our liver constantly produces it, sticks it into the plasma, and it's just floating around. All right, so there's a number of things that can cause the release of renin from JG cells. Remember, JG cells are the juxtaglomerular cells, and an increased sympathetic stimulation can do it, changes in the macular density can do it, or a drop in blood pressure. But any of those things can cause the JG cells to produce renin. And now, renin's in the bloodstream. Angiotensinogen is getting floating around in the bloodstream. And when they run into each other, it cleaves off a couple amino acids and basically turns this inactive form of the hormone into angiotensin 1. Okay? Now, angiotensin 1 is an important hormone in that it's not very active, it's very wimpy. But that then, once we do that, as it moves through the blood vessels and the endothelium, remember the lining, endothelium is the lining of the blood vessels, especially in the lungs, but also it seems in the kidneys, but the lungs is a big place. We have this special enzyme called ACE. The ACE enzyme stands for angiotensin converting enzyme. So the capital ACE, angiotensin converting enzyme, and it's embedded in the walls of the capillaries in the lungs, and there's pretty good evidence that there's some in the kidney as well. And so as angiotensin 1, that's in the blood now, in the plasma, flows through the lungs, it gets converted to angiotensin 2, so we see AG2 down in this box. And angiotensin 2 is the really, really, really potent form that has a lot of vasoconstrictive activity. Just as an aside, right, it seems like one of the ways that the novel coronavirus, right, the one that's causing pandemic, right, is attracted to the receptors on the angiotensin converting enzyme. And so that's certainly right now a huge level of investigation going on around that area to see if there's anything we can do to lessen that. And it's that binding to those receptors there that causes the inflammatory response and typically the body to overreact. And that causes what they call a cytokine storm, where we produce a lot of these chemicals in the body, and that causes pulmonary edema and obstruction of the lungs, and is the big thing we worry about for people that have to go on a ventilator, and eventually could even die because of that. So it does have a role in what's currently going on in the pandemic right now. So what does angiotensin do? Well, let's kind of look at the name. Angio refers to vessel, and tensin is what it sounds like, tension. So it's a vasoconstrictor. That's what angiotensin means. So its major action is vasoconstriction. Okay? Its minor action is fluid retention. All right? So it also helps to retain fluid. And the reason it does is because it is such a powerful vasoconstrictor that it constricts the afferent arteriole. And as it constricts the afferent arteriole, it reduces the filtration pressure within the glomerulus, which reduces GFR. So if I have less fluid going and being filtered into the kidney, that means I have more fluid everywhere else in the body, so it effectively increases blood volume. In addition, it also targets the hypothalamus. So yeah, it's got the vascular smooth muscle receptors where angiotensin 2 binds to, but it also affects the hypothalamus to increase the ADH production, which helps support everything it's trying to do. And it seems like the rate limiting step, the step that really makes a difference in the speed of how this enzyme works, is that renin. Okay, and so that's the rate limiting step, and it's the more renin we have, the more angiotensin 2 we have. So renin is stimulated by a decrease in blood pressure. So a direct decrease in blood pressure will cause less stretch of the JG cells, and that would release renin. The macula densa, through the tubulo-glomerular feedback mechanism, can sense changes in the tubules or the arterioles and initiate that response. So that's another way to change renin. And we also talked about the ANS, which is sympathetic only, and sympathetic stimulation directly causes a release of renin. So those are the ways that we can change the amount of renin we have, which ultimately affects angiotensin 2. The other thing, just to kind of mention off to the side, is since the ACE receptors are actually a good place to pharmacologically affect people with hypertension. So one of the more common drugs now that people take for hypertension is what they call ACE inhibitors. So blockers to this ACE enzyme, right? And if we have less ACE enzyme, then we get less conversion of angiotensin 1 to angiotensin 2, which means that we have less constrictive effects, which means we lower blood pressure. In addition, there's not really widely available, but they're actually working on another type of blood pressure medication that would affect the release of renin from the juxtaglomerular cells instead of affecting the actual action of the enzyme, angiotensin converting enzyme. So, that's kind of essentially what happens. And we see that the two major effects then are vasoconstriction everywhere in the body, which increases TPR and helps to increase blood pressure. And then the big effect on the afferent arterial, which then decreases GFR and effectively raises the blood volume. Now, those were our 3 water conservation hormones that helped us to keep water high in our body, not high, I guess at a normal level, and that helped to maintain blood pressure. So now we're going to talk about the one that does the opposite, that's antagonistic to the other 3, and that's called atrial natriuretic hormone. Sometimes it's called ANH for atrial natriuretic hormone. In the past, people have argued about whether it really was a true hormone because it's pretty small in size, and some people argue to be a protein hormone you had to be so big and it didn't meet the criteria, so people started calling it a factor, or ANF is sometimes used, and then people said, well, not really a factor should be, and they argued about that, and then they called it a peptide, so you see ANP. So if you see the word I guess the acronym ANH, ANF, or ANP, it's typically talking about atrial natriuretic hormone. So make sure you understand it's 3 different designations for the same hormone. It's actually, like some of the other ones, a fantastic name if you know how it works. So I think most of you realize atrial, right? Tells us where it came from. Okay, so it took a long time for scientists to figure that out because nobody really thought about the heart being an endocrine tissue as well. But it is, obviously. And natriuretic is not as hard as it seems initially. So if you kind of look at the uretic, right? So a diuretic means what? You pee out stuff, right? So uretic is pee out, basically. And the natri. So I think if you thought about it for more than a second, and some of you probably already came up with it, but what's the chemical symbol for sodium, right? Well, it comes from, our Greek and Latin words natrium. So the natri, right, refers to sodium, and uretic means to get rid of. So this is a hormone that gets rid of sodium that comes from the atrium. And really, it's a great name. So the source, like I said before, is the atrium. Tends to be more dominant in the right atrium than the left atrium. And that's because that's more reflective of the systemic blood pressure, right? The left atrium gets its blood from the lungs, but the right atrium gets its blood from the rest of the body. So if there was an excessive blood volume or pressure, then that would be felt in the right atrium. And if the right atrium had excessive stretch, then it would release ANH, and that ANH would help to decrease the amount of sodium in your body, and since we peed out more sodium, water would follow along and would help decrease blood volume and blood pressure. So it's the amount of stretch in the atria, and especially the right side, that really determines how much ANH is released. Its basic action is to block sodium reabsorption. So if you remember from aldosterone, we said that aldosterone increases the activity and number of sodium reabsorbers. But in this case, right, the basic action of ANH is to block these receptors. So instead of pulling sodium back into the body, right, what happens is we leave it in the urine and it gets peed out. So that's called a natriuresis. So the target is the ascending limb of the loop of Henle, and it literally is the exact opposite of aldosterone. It decreases the number and or activity of the sodium reabsorbers. So this is a good guide here, and you'll see a similar similar type of table likely posted on Canvas for you to study from. I think it kind of puts everything in a nutshell and helps you to kind of figure things out. In addition, for our group problems, it's likely you'll see these hormones there as well. So certainly understanding these hormones and how they function, an important part of this. I'm not going to do the summary here, but you can kind of read it and figure that's pretty important. So that's the extrinsic regulation of GFR, and we're not going to worry about any others. So now is probably a good time to stop this one and then just pick up with clearance and a few other odds and ends on the next video.